Hello and welcome to another thrilling episode of Biopsychology. We're going to be looking at Chapter 6 today, which is on vision, probably one of the most amazing aspects of our biological makeup is the human eye. Very acute, very binocular, very high up, all these evolutionary components that make our eyes an incredibly important aspect of our survival. So let's take a look at the biology involved in here as we explore Chapter 6 on vision. We start off with, generally speaking, in terms of the sensory systems, all of them that we're going to be exploring in this class operate in the notion that there are energies outside of the body that undergo a process called transduction, where energies from the outside are transferred and picked up by sensory neurons of some kind specialized with sensitivities to those outside energies, and these are eventually translated into electrical energy, which is then translated as the nerve impulse into the nervous system in order for us to process that in our brains. Specific to the vision system, we have all the light within the visual spectrum, which is just a very small part of the electromagnetic spectrum, which includes everything from gamma rays and X-rays and microwaves and radio waves and all that. Depending on frequency and amplitude, there's only a small part of that we're able to see with our eyes, that our sensory neurons are specially tuned within the retina to pick up on those particular wavelengths and translate those through the visual pathway, which we will examine with some mapping of the brain in terms of what happens to this information. Now, of course, it all starts with the most amazing of organs, and that is our eye. Our eye specialized in its function, in its form, to receive light from the outside world. All of its pieces work together. We have the cornea and the space, the anterior chamber right on the outside of the iris and pupil. The iris constricts in order to let less light in. It expands in order to let more light in, primarily focusing incoming light onto an area called the fovea, which we'll talk a little bit about later. But the fovea is a concentration of cones and the cellular parts of our retina that is the most in focus. So the only place in our retina where things are actually processed to the acuity that they're in focus is at the fovea. So if you look at any part of this diagram in here, you look at the word pupil and you just pay attention in your peripheral vision, and you'll see that the rest of the words around this diagram of the eye are all a little blurry because they're not focused on the fovea. If we move our eye to any other aspect of this graphic, that little piece of the graphic comes into focus as our eyes move around and maintain that light directly on our fovea. And we'll see that when we talk about some of the movement of the eyes that allows that to happen. Light, of course, is coming in from other directions and activating cells along the interior of our eye. This is more of the retina, but those particular areas in our peripheral vision are more attuned to sources of light and sources of movement. So movement off in our peripheral vision will stimulate those cells more than movement when we're directly looking at. In fact, let me I was reading a book once, and it was about tracking animals and how do you see animals moving through a tree if you want to find a bird that's in a tree. And in the book, they introduced a thing called spider vision. Not Spider-Man, none of that. But spider vision was to focus in a space when you're looking up at the tree where you're not focusing on a leaf, or you're focusing on a blank space in the air. So everything seems blurry. So you kind of practice this, setting your focal point to being on a non-object. And what happens is all you all your eye is completely paying attention to peripheral vision because it's really not focusing on anything. And any movement in the peripheral vision, you pick it up right away. It's actually really, really interesting to do. And it kind of relates to this physiology of movement in the periphery. This is adaptive. When I'm looking at something, if something's coming at me this way, I want to have really, really fast neurons to tell me something's coming this way at me or this way or any way while I'm focused on what I'm doing. When I'm focused on something, the movement of my eyes will keep that thing in focus. I'm tracking that thing with the movement of my eyes while my peripheral vision is tracking the movement that is outside of that. So very cool system. Very cool system. There is a space, however, where all the axons of all the receptor cells are going into and create what's called the optic nerve. And at that particular place, we call that the optic disc or the blind spot. You've probably heard of that before. And there are no receptors there. Doesn't pick up on information. And we can actually demonstrate this with the blind spot experiment, the first of the illusions that we have. It's kind of an illusion, but I can help you to make one of these things disappear from your actual visual system. Now, the best way to do this is actually to look this up, print that out, and actually hold it in front of you. And what you do is you close one eye, and as you're looking at it, you focus your eye, put the black dot on your fovea. So you're just looking directly at the black dot, and as you move the paper forward and backwards, there's going to be a place in which the cross will disappear because that light is actually shining through your eye from your peripheral vision and landing on the blind spot, and then nothing's getting picked up. So that little X will disappear. Very, very cool. You have to close one eye because binocular vision, because something that's in the fovea on this eye, now something that's in the blind spot on this eye may not be in the blind spot on this eye, so there's no blanks when we have binocular vision. But when we just have one eye, then we're susceptible to that blind spot area. Okay, so very, very neat way of experiencing that is to print this kind of thing. You can actually do it on the screen. I'm doing it right now. I'm leaning into my screen and I'm looking at this is what I look like when I'm doing it. I'm going like this. My one eye closed and I'm looking at my screen, and right now the cross it's not within my vision. It disappeared. It just looks white over there in my peripheral vision. As soon as I focus on it, it reappears. Now let's take a look at the cross section here of the actual retina itself and all the different cells that make up this inner coating on the eye, this very, very rich cellular area that makes up the retina, which is the photoresponsive part of our vision system. Now there's different layers here. First off, let's get this right. The light is coming from the left. This is that orange area is the inside of the eyeball. Okay, so light is coming from the left, and you'll see that what it sort of hits is the axons, all these axons from the retinal ganglion cells, which we'll talk about next, translating information from the inside of the retina all to that blind spot and creating that optic nerve that's going to go to the back of the brain into the optical cortex. And so the light is coming from there. It passes through and around these axons and comes into contact with these. And it sort of explain what each one of these cell layers means, because these initial cell layers are actually communication cell layers. They're not actually the ones that are responsive to light. It's the rods and cones are responsive to light. That's where the transduction happens. But all the light passes through these other cells. So by the time it gets to those rods and cones, it's had to work its way around all of this cellular tissue. It's interesting when we talk about perception and how we regroup that information all together. When the actual information that's sent to the occipital lobe, is rather distorted because this light is going through all of this cellular tissue to finally make its way down to the rods and cones. But nonetheless, we have ganglion cells. They receive from the bipolar cells, we'll talk about in just a little bit, and these are the cells that send the image to the brain. They send the information down the axons of the retina to the optic nerve and then into the brain through the visual pathway. The next set of cells that we see here are amacrine cells. Now, these are crosstalk cells, and what they do is they analyze the information that's being projected onto the entire retina, sort of a large, like a bigger picture sort of thing. And what they'll do is they will release chemicals into the rods and cones areas to suppress their reaction when the eye is trying to focus on a particular area. So there's actually a suppression of focus, there's a suppression of sensitivity, there's a suppression of brightness because our eye is focusing in that fovea area on the place that we're of most interest. So not only is that peripheral vision that we have around ourselves out of focus, but it's being actively suppressed by the amacrine cells. And this can be experienced very, very clearly. This idea of suppression can be experienced with this particular optical illusion. So I put this graph up here, and as you're looking at these boxes with white lines intersecting all over the place, if you focus on any intersecting white line, what you might experience is in your peripheral vision, it seems like there's like a little grayness that appears in all the other intersections. And what you're actually experiencing is the effect of the amacrine cells suppressing the cells around the fovea, reducing the brightness of those particular cells. The black ones, obviously not a big deal, but reducing the brightness of where these places cross, and you get that little shading. And this is kind of a way to visually experience the impact of the amacrine cells that are actively suppressing around the place that you're focusing on. So you see that intersection very clear. It's in clear white. You look back at this and see, if you look at any one of these intersections, all the others seem to have like a little gray spot in them. As you move around, the gray spot, of course, they move out of the way as your amacrine cells are actively not suppressing these cells of where you're looking at. So kind of a neat way of experiencing that particular aspect of our neurons in action. Here we have the bipolar cells. And no, these do not have any sort of mental illness. They're bipolar because they send and receive information both ways. There's a sort of they're a translator neuron connecting the inner cells of the retina to the outer cells. It is interesting that there's another level of processing here, a little mysterious as to what this processing actually is, but this is the only sense of all the senses that we have. Think of hearing, taste, balance, all of those other ones, hunger, all those different senses that we have. This is the only one that has that intermediary, has one other level of processing going on, refining the message. We don't really know, but there's this messenger that's in between the inner parts of the retina and the outer parts. So an additional layer of processing. Whoops, pressing the wrong buttons over here. Then we have horizontal cells, and these are interesting in that, similar to the amacrine cells, they take a larger picture, but they do this by not by suppressing neurons, but by creating a generalized feel for the picture of what's going on. And I can only explain this sort of with an example. In this, when you do video, digital video, there's a processing of light information that takes each color in the image that you're looking at right now and processes it in order to turn it into a different kind, to turn it into an actual, from a photon into a signal that can be understood by a computer, similar to the eye. That's what cameras do. Now, they're called codecs, and they're computations of different colors. And some codecs are designed to take a certain range of colors and sort of generalize them, because if I can generalize that color, I don't have to keep as much information going on. So you see this green background around me. And in truth, the codecs that I'm using are not really sophisticated. It's not creating a completely plain green black background. But I'm going to tell you that if I turned around and looked at this background, which is a painted wall, I can see a lot more variance in color than you do, because similar to these horizontal cells, the computer is taking all of these different greens and it's kind of making maybe 3 or 4 greens out of all this, which is easier to store in memory, doesn't take as much room, and generalizing those. So some of the detail is lost, some of the different detail. But if I look at it, I see all kinds of variations of different greens because there's no codec. There is a codec in the way I'm just talking about that, but it's not quite as generalizing as this. We want our eyes to see that kind of detail. But in this kind of video, you're only seeing maybe 3 or 4 different kinds of greens here. And there's actually probably 50 different kinds of greens if I looked around. Just slight modifications in the light and the surface texture of the paint, and stuff like that. Computers do that to limit the amount of information. The same sort of process is happening here. And that can lead to some interesting optical illusions. And we can see what can happen is we can lose some detail in our visual system because the horizontal cells are kicking in and making some generalizations for areas. And they prove to be some very interesting optical illusions that can come from that. So let's take a look at one of my favorite optical illusions, and that is the gradient illusion. Now, I've created this on a computer, but there's no trickery here. And what you'll see is on the left-hand side, you have a graphic that is a gradient from black to white. It gets over the course of going from left to right, the colors become increasingly white. And you can see the resolution here. You can see lines even, because a computer can only generate, a certain probably 75,000 different colors or 75,000 different shades of gray, whereas our eye can pick up on billions of shades of gray. So in the real world, we see much more smooth gradients here. I then copied a little piece of that and I created a little version of this over on the left-hand side. You can see a little square that also has a gradient. Now you can see that that little square on the right has slightly darker on the left, and then it's actually a square, and it actually, it gets lighter and lighter until it's totally white and it blends with the background. Now here is the effect of our horizontal cells. As I move this image graphically, move the little square over to the other side, watch what happens to the little square. So it's moving over to it's going to disappear. And then once it goes over to this dark area, the gradient is overwhelmed. The gradient that's inside of that square is overwhelmed by the darkness around it, and the horizontal squares turn that square all into one color. White. So our eyes see only one color this time. That's the same square as this one, which has a gradient. That gradient will disappear as we change its background, and the horizontal cells say, okay, there's not enough difference between the left and right in this square to really pick up on a gradient. I'm just going to generalize this as one color, and it's almost impossible to see that the left side of this square is actually a different color than the right side of this square. So we see this in a classic illusion right here. I actually have a paper version of this one in my office, a large poster where I can demonstrate this. And what I'm going to show you, for the same sort of generalizing ability that horizontal cells have on colors and shades of gray and whatnot, I'm going to convince you that the color of the block A is the same as the color that has the B in it. These are the exact same color. Now, it doesn't seem to make sense because the B seems to be just one of the white squares in shadow, but the A is the same color as the B. And again, I'm doing this graphically, but there is no trickery in here. I've sliced a little piece of this, of the graphic from A, and I'm going to have it move into B, and you'll see this little square is going to move. See right there, that's out of A. I'm translating it down here, and I'm going to move it into B, and it disappears there. So those are the same colors. That little piece is that I didn't have it change color along the way. That's why the paper one is really interesting, because I can actually take the little square and I just move it physically right on the, and it literally looks like it's changing color, but it's printed. So it's just going, we're not in Harry Potter land here. It's not changing color, but the relative area around. Again, this is our horizontal cells kicking in for some generalizations that allow us to do this. Now I'm going to go ahead and move that square back from being in the B square, move it back up here. It almost seems to change color right there, but it's actually the color of block A and the color of block B are actually the same in this image. The last one I'm going to show you is important. It's an important quality related to the way in which the eye processes information and makes generalizations using these horizontal cells, and how important it is for camouflage. There's actually an image of a heart in this, like not the anatomical heart, but the little heart. And you might have a sense of where it is right now, but because all of these dots are all mixed together, it's really hard to pick out where that image is right now. However, if I move them all together, I introduce movement, and movement starts to move, a bunch of these dots are going to start moving together. It's very easy to pick it out in terms of its background, and then it's going to settle in again and probably disappear again. Even though you're watching it right to the end of its movement, as soon as it stops moving, it disappears into the background. Again, the generalizing quality of our horizontal cells. So here we have, it's moving around. You can clearly see the heart even goes out of bounds here a little bit. It's going around and it comes down. It's going to settle down here pretty soon. See if you can keep an eye on it. And you know it's there, but where did it go? Which ones were involved? And so it disappears again. And here we are seeing again that horizontal cells making generalizations about the whole field, and how that results in us seeing things in certain ways, and the ways that our brain generalizes what we're seeing. It essentially to kind of reduce the amount of information that we have to process when we're processing something visually. Okay, let's continue our journey down the retina. And our next part here, we are in our cones. Now cones, we have about 7 million of them per eye. This is high acuity. This is where very, very clear vision, color, and day vision. And these are concentrated around the fovea. Cones exist all over the retina, but they're concentrated in the fovea. So the eye focuses light on the fovea where we can pick up high acuity color and our day vision. That's what we want our vision for. The rods, on the other hand, lots more of these that cover all of the interior along. They're in the fovea as well, but not as many as there are in peripheral vision. 140 million of these, lots of them per eye. Night vision, light detection, movement, those kinds of things. That's what those trigger. Okay, so that's our peripheral vision. Now we can experience this, for example, when we look at the night sky. Now this is really not a good example because this is just an image, and so almost all of the points of light that you see are equal in intensity, and your eye will process them because this is not a real image. However, just last night, and I would encourage you to do this, go out and look at the night sky on a nice bright, I was gonna say bright sunny night. No, go when it's really dark. Not, you're out in the field someplace, no city lights or something like that, and you look up to the sky and see if you can find a satellite that's going around. Sometimes you can see a little dot. It's not a UFO, it's actually a satellite. And, well, if it is a UFO, that's awesome. But you see something going on. And here's the interesting thing. If you try to follow it by focusing your fovea on it, it tends to disappear because in your fovea, there are not enough light receptors for that dim light to stay in there. What you have to do to see a satellite is to move your eyes slightly to one side or the other and pick up on the movement. If you look directly at it, it seems to disappear. So when we're looking at the night sky, and astronomers do this, particularly early astronomers that weren't, when they're looking with the naked eye or we're looking just through telescopes, that the peripheral vision is actually a better part of our vision spectrum to pick up on faint light. We can see more stars in our peripheral vision than when we particularly focus on them, because when we focus that particular light, having traveled millions of years to get to us, that particular photon will probably not fall on a rod, it will fall on a cone. Not a whole lot of color variance, not a whole lot of movement, it's going to disappear. Whereas if we kind of move our eyes slightly to the left or slightly to the right, we'll see that movement, for one thing, just like we see with that kind of spider vision I was talking about before, and we'll tend to see more. We'll see. So, if you kind of just look at the dark spot in the sky and just wait, and then you're going to see movement you might see movement in your peripheral vision, and that's when you can start tracking the satellites going by, or you'll just see more. You'll see more stars actually in your peripheral vision. So very, very cool stuff. Now moving away from the retina, now we're going to look at the function of the eye, a little bit of the movements of the eye that actually allow us to focus the light on particularly the fovea and move the peripheral light to the cone, to the rods that we want them to be focused on. So eye movement itself, so that we can track information and we can move our eyes as objects move, and that we can move our eyes as we move. And so let's take a look at eye movement and the mechanisms of eye movement. And I want to give you a demonstration here of the first kind of eye movement, and that's when we're trying to look at objects like this. Now, if I asked you to look at these circles, that you focus on the circle number 1, and then going from one circle to the next, all the way to 9, you focus on each one of these, but try to do it really smoothly, like going from one to the next. And as you try to move to the next one, moving your eye, what you will find is it's nearly impossible to look at the next one and the next one smoothly transitioning over. You tend to jump to it. So when you're starting at one, and you jump to the next one, and you jump to the next one. And even if you try in between, you're just like, it's kind of like this jerky movement as we're looking at the static objects all the way over to the 9. And this is what's called saccades movement, or saccadic movement. It's this twitching of the eye that moves from object to object. And as we're looking around, we don't tend to do this smooth vision. We tend to like look over, and then we're looking at that, then we're looking at this, and it's immediately this kind of jerky motion as we're moving our eye around the field of vision. Now, alternatively, that if something is moving within our field of vision, it's much easier. So let's say, for example, I want you to follow this. Keep your head still, just look at this spot, and I'm going to move at the same distance as from 1 to 9, and the spot's going to move, and your eye is going to be able to follow it as it makes its journey. Now, depending on the quality of the video, you might be a little bumpy along the way there, but pretty straight. Your eye is able to smoothly follow that just as it's going across, your eye movements are there. When you tried to imitate it on the ones on the top, you just had that jerky sagatic movement. In this case, what you have is smooth motion movement, I think. Now, I can move this all over. Keep an eye on this thing. It says it's moving all over, and your eyes are following it, doing its little journey around over here, all over the screen, and comes back up here. And you had no problems. Your eyes weren't jerking around, looking at this and that. Smooth pursuit movement. Sorry, I got that wrong. Smooth pursuit movement. So there's two kinds of muscle movements in your eye here. Keeping your head still, you had the saccadic movement going from object to object, and then at the same time, same set of muscles doing smooth pursuit when the actual object is moving on its own across your field of vision. Now, in addition to that kind of movement, we also have, here's our two eyes, and we're looking at an apple. Now that apple is situated in front of us, and we position our eyes so that the light waves are focused directly on our fovea. As the object moves, the eyes move in coordination with each other, and they keep the light focused on the fovea. No matter where that apple goes, as it comes in close, we get kind of cross-eyed. As it moves out, our eyes adjust. And this adjustment of movement, you'll see sometimes they're moving in opposite directions. As the apple moved to the left, they both went to the left, to the right. But as it came closer, they converged. And as it moved out, they moved apart from each other. This is called vergence movement, which is the eyes coordinating their movement to make sure that both eyes are receiving the information right on each other's fovea. Now, an interesting aspect of this is that vergence is one of the ways in which we understand depth of field. I mean, how far an object is from us. If you think about, if you put your finger out in front of you and you look at your finger, like this, and you bring it closer to you and you keep your eye on your finger, eventually you're going to really feel some muscle tension on the outside and inside of your eyes as the eyes are focusing closer and closer. And as the object gets further, the eyes depart from one another, and pretty soon they're looking pretty much straight ahead, and there's less tension. Our sensing of that tension is one of the signals that our body uses to determine whether an object is close to us or an object is far from us. So this is part of our depth perception. We have this sort of tension on these areas, these muscles that tense up and then release. Where I put these red lines, you can see where these are. That muscle tension is another one of the cues that we have to living in a three-dimensional life. Okay, the last one I'm going to look at here is what happens when your head is moving. So here I put the apple again on the screen. I want you to focus both your eyes on the apple and shake your head like you're doing no, but keep that apple right there. Like you're doing this, but you're keeping the apple right in your vision. And it's sort of like smooth motion, except I'm moving, not the apple. Okay? So I'm shaking my head now. In fact, you can move your head all around. You can swirl it around. You can do figure eights and stuff like that. And you can keep that apple right on your fovea that whole time, no matter how much you're moving your eyes. That's called vestibular ocular movement. Now, there's coordination between the vestibular, we'll get into that, the vestibular aspects within your semicircular canals in your ear that allow for spatial relations along 3 dimensions. And so when I tilt my head like this, my brain, because its response to gravity, is realizing my head is now at a 45-degree angle this way, and my eyes will accommodate to that because information from that vestibular system is telling me I'm 45 degrees off. Does that very quickly, and the movement of the eyes willfully can keep it focused right on that object. That's gotta freak you out. That's like, wow. Really cool stuff. All right, let's go into the visual pathway, our last major area that we're going to go into today. And this is probably the most studied of all the pathways in our brain, the most understood, but yet still largely misunderstood. Let's take a close look here about what happens. We have information from across the visual field, in this case represented by this girl's face, and the numbers 1, 2, 3, 4, 5, 6, 7, 8, 9, depicting the different parts of this picture that are being translated. And you'll see some parts are going to both eyes, some are just to one eye. You know, that depending on where these things are at, there's always that kind of a duplication because we have binocular vision. But in a sense, we have information from, let's say, this left part of the image coming down into both eyes. And you'll see that some of the light hits the inside area of the retina, and some it's the outside, you know, that depending on which aspect of this picture we're paying attention to. We have the same thing going on the right-hand side, information coming from this picture translating into both eyes. And then we have the movement of that information. We have information now that's been transduced into electrical information on each eye, and we can see probably something from your intro to sight class. We see that we have two hemispheres, and information that is processed that's coming from the right part of our body is processed in our left hemisphere, and information comes from our left side of our body is processed in our right hemisphere. This is not completely that way. This is an interesting, you know, visual pathway coming into this area called the optic chiasm that I'll demonstrate better in just a second. But in essence, we have information going from one side to the other. Now let's take a closer look at that particular pathway here. We'll look across from the image all the way to that part of the brain. I want you to recognize when we follow the photon. Now we have a photon of light up here in this part of the image. Now I'm going to follow one of the pathways. That area is making itself known across the whole retina here, but let's follow this one photon. It goes into the eye and it activates that area of the retina. Once that transduction happens, that information is sent and it travels along the visual pathway to the optic chiasm. And in this particular case, because of this particular location of the retina, it does cross the corpus callosum and translates from one hemisphere to the next, and it goes over to the primary processing visual area here, and it's translated to the cortex for processing of that particular piece of the picture. Now the next photon is near the number 7, and that photon, the one I'm going to concentrate on these over here. This one goes over to this eye, way over to the outside of that particular eyeball. Now that information gets translated to the optic chiasm, but it doesn't cross over. Some of the information stays on that side, it's processed over here, and it goes to its appropriate area for processing on the visual cortex. Now what's interesting about the visual cortex is that the visual cortex is mapped. You see this yellowish area of the real world, and it is reflected in the mapping out of that image on the actual surface of the cortex in very much the same way. The information is, of course, reversed and upside down the way it's being processed, so it's an upside down image on the back. But the actual nerves corresponding nerves on the cortex that are next to each other correspond to neurons on the retina that are next to each other. That information comes together to create a neuron-based image on the back of the brain. There's very, very fascinating sort of process there. Here's a really interesting bit of information about that too, is we often think that the brain in development, we often think of the brain follows the courses of evolution, meaning that the functioning areas of our brain having to do with vision are stimulated into becoming vision neurons when light information starts coming along. It's like you're let's say you get hired as a clerk in a store, but you're not sure what you're going to sell. So you're waiting for the customers to come in, and all the customers are asking for Pepsi, so you become a Pepsi dealer. So maybe that's not a good analogy, I'm not sure, but everybody coming in is asking for Pepsi, so you become a Pepsi dealer. The environment impacts the function. Okay. The interesting thing about the development of the visual pathway is it actually develops similar to the way we build industrial bridges. We actually start on both ends and meet in the middle. So developmentally, this is a kind of a fascinating part of this. The visual pathways are predefined in the occipital lobe and predefined in the retina before they meet. And they're looking for each other and match up. It's not a one-way trip and then they get created. They've been created and then they meet each other when they create the optic chiasm. I hope that blows your mind as much as it does mine. Okay, let's look at the visual pathway. Here we have looked at information coming in to the eyes, passing through into the optic nerve, crossing over at the optic chiasm, being distributed across the visual cortex. So here we have, right from the course book here, we have the bear, and that's where our photon is. We're going to take a photon from the bear. The bear photon comes into the brain. This straight line is actually that visual pathway we just looked at. So the photon comes in and it reaches the visual cortex through the processes that we've already discussed. Now we're processing information. How do we actually learn about this bear, where and what it is, and determine what we're going to do when we have this, the actual response when we see these things, the very function of why we want to gather this information in the first place. So part of the information being processed here in the visual cortex, represented by this little black dot, gets translated through the dorsal stream, which is one of the visual pathways, into the MT area of the visual cortex. Now, this particular area of the visual cortex, kind of nudging up against the parietal lobe here, where we see the motor and sensory functions, this has a lot to do with direction. This area processes direction, essentially giving us information as to where this thing is. Where is the bear? Is it in a cage? Is it way across the river? Or is it right next to me? That kind of thing. Because all that information about depth of field and how far is it from me and all those things, that's the processing of where the object is. At the same time, additional information, represented here by an additional black dot, is getting processed and sent to the V4 area, lower than the MT area of the occipital lobe. This area has a lot to do with attention. A little bit toward, the kind of the base aspects of the brain here. The information is translated along this pathway, activating the limbic system. This is our emotional responses here. We're not even quite sure what this is yet. Not sure if it's a bear. It's starting to look a little scary, and nonetheless, we activate the limbic system, and that information, that emotional response, is sent to the prefrontal cortex for processing. The signal carries on into the ventral stream, where we add language, words, information about objects, and we're processing this even more. Now we know what it is. Now we know what it is and where it is. We know it's a bear, and we know it's close. That information is subsequently, through various pathways, sent to the prefrontal cortex, and this is where we decide what we're going to do. So this is how information is translated using the entire brain in order to make a decision as to whether or not we're in danger, activate the right systems. I think it's really cool that on the way toward understanding what it is, we actually activate the limbic system, so our flight or fight response is activated. And you know this because sometimes you come to the realization that It's only a stuffed bear. But your limbic system has already been activated. You're already worked up. You were scared for a moment, but now it's not real. So, even as you were making the determination as to what it was, your limbic system had already kicked in emotionally. Fight or flight had already started a little bit. And so even though in the end you realized it was a stuffed bear, you're still like, whew, that was close. I thought that was real. That's cool. All right. So that brings us to a little bit. I've given you some examples I want to talk about because it's part of the assignments, optical illusions. You saw throughout this presentation, I used various examples of optical illusions and explained why they happen as a function of either the structure and function of the retina or of the processing. You say it's illusionary, if we think about it, that a stuffed teddy bear can activate my fight or flight response. But that simply demonstrates the course and order in which information is processed in the brain. And so even though the fear was there, it was due to an illusion. Okay, so those count as well. So I'm going to be asking you as part of the this week's work to go and look for a visual illusion of some kind and see if you can find a source that will explain the actual neurology behind it, or take a stab at it, understanding the way the nerves work in the retina. If you can look at something and see if you can figure out why that particular illusion works, or that that optical illusion, and see if you can figure that. So that's one of the assignments. You're going quickly to the assessments. Here we have, of course, the discussion. This is in the discussion. Locate, get a picture or a link, and explain the optical illusion. Why does it trick us based on the nervous system? There's a quiz on the visual pathway. We're actually following some of those photons again. And we have an assignment, a brief assignment, just a little research paper, very, very small, 2 to 3 pages. Title page, references, all that stuff. You're all psych students. On an animal's vision, on some other animal, be it a whale or a dog or an eagle eye, they got pretty good vision. Those kinds of things. Just doing a little bit of research on another animal in the world and its vision system and how it compares to the human vision system. And that's it for this chapter. So I look forward to seeing all that work, and have a great week.